Hello. In this video, we're going to discuss the last of the electron transport complexes. So finally, we're going to see all these electrons, that we keep bouncing around, that we got in the form of an ADH from TCA cycle, and then went on from complex 1 or complex 2 to reduce coenzyme Q, and then from complex 3, they reduce cytochrome C. So now we have cytochrome C, that the iron in cytochrome C in the heme there is reduced. It's iron 2. And finally, complex 4 here will take these electrons and pass them on to oxygen to get water. Okay. And in the process, again, we have translocation of protons from the inside of the mitochondrial matrix to outside in the intermembrane space. Complex 4 is also called cytochrome oxidase, or COX short, because of course it oxidizes cytochrome back to the iron 3 form of cytochrome C. Now, we see a lot of protons coming from the outside, from the inside, 8 of them. Now, only 4 of those protons will actually be translocated outside. The 4 of them from the inside will be used to reduce oxygen to water, right? So the scheme, the way it goes. So here we have prosthetic groups that have iron in cytochrome C and copper in complex 4. So there are 2 of those, copper A and copper B, of these prosthetic groups that are copper dependent. Of course, the complex itself has other hemes and so on and so forth, eventually making it all the way to oxygen. So, move myself back up again. This is the outside, and the electrons coming from the cytochrome C. Remember, this is a cytochrome C is on the aqueous side of the inner mitochondrial membrane towards the cytoplasm, towards the intermembrane space, and the protons all come from the matrix. All right. So the electrons hit copper A, and then they go through the heme A here in complex 4. And before all continuing on to copper B, and other hemes here are involved, A3 heme, to finally reduce oxygen that comes in here into water. So, and in the process again, 8 protons play a role. 4 of them will be translocated, and the 4 will be used in the reduction of oxygen. Now, we saw 4 electrons because, as you remember, you need, or you may remember, we had discussed earlier, you actually need 4 electrons to reduce oxygen into water. Okay, so they are important. Exactly how this happens is not clear. This is the latest scheme from Leonid Sazanov's lab, again at the MRC. This is the same lab that showed the high resolution structure of complex 1, which is the best understood in terms of how the protons are translocated across the inner mitochondrial membrane. You see the 4 electrons coming in from cytochrome C at the 4 steps, okay, and the involvement of how oxygen that comes in at step 3 here, how it goes through different forms until finally we get water. Okay, so this is the best scheme that we have for now. We don't really understand how the electrons are translocated. So unlike the situation of complex 1, which again is the best understood, here we don't really know. There are some channels that the Sazanov lab again has made some predictions. So I'm sure at some point in the future, when they have really high, several, not just one, high resolution structures of this complex, they will be able to make a precise model of how the protons are translocated. Again, the whole idea of the electron transport is, let's go back 1 or 2 or 3 slides. As the electrons move through all these cofactors, remember we discussed how in hemes the iron, depending on how many unpaired electrons you have on the iron atom, it gets bulkier or not. So the more unpaired, the bulkier it is. And that triggers conformational changes. It's these conformational changes that enable protons to be translocated across the membrane. There are mutations that affect that are observed in people that map onto complex 4 function. And of course, they lead to diseases, very serious mitochondrial diseases. Again, there is exercise intolerance, inability to breathe, and so on and so forth. And most affect the assembly of the complex, similar to what we had seen with complex 1. It's a complex procedure to put it all together, complex 4, and assembly factors are needed, and it's usually mutations in those factors that are recovered in patients, and we see. And that concludes our discussion of the whole electron transport chain. All the electrons now, finally, we can see how they end up on oxygen. And all we got so far are protons, moving from the inside of mitochondria to the outside. Okay, so now we have charged our battery, but we have not seen any ATP or anything like this. Okay, this is just the electron transport assembly here. So we'll stop at this point.